All right, well, welcome everybody. We're going to get started here. It is 4 o'clock. Welcome to another installation of the Crow Canyon webinar series where we have Jonathan Dombrosky today, and I'm really excited about this presentation. I want to go through our intro slides and then introduce Jonathan, and then we'll get started. First, I want to thank all of you who are joining us today for helping make this possible. If you weren't here, and we've got well over 100 people, 120 folks who've signed on so far. If you weren't here, we wouldn't be doing it. And we've really gotten a lot of support from this community to continue this webinar series throughout the COVID pandemic. So thank you so much for supporting socially distanced programming at Crow Canyon. A special thanks to Dylan Schwent, Taylor Hasbrook, who have been organizing this whole thing. Jonathan shaking his head, he knows Taylor's just really organized. She's got this down to an art. And we've also received support from the National Endowment for Humanities, the Colorado Humanities Cares Program, and the Region 9 Economic Development District in Colorado. These are 3 programs funded through the CARES Act that was passed by Congress this spring. So thank you very much. We also want to thank the people whose land Crow Canyon is on. Specifically, we want to acknowledge the Pueblo, Ute, and Dene people on whose traditional homelands we work and reside. And we're grateful to all Indigenous people who continue to preserve and protect cultural heritage across the Southwest and around the world. And, you know, this has been a really challenging time for some of our Southwest Native communities. Through working with our Pueblo advisory group at Crow Canyon, we've identified some nonprofits where you can help. And they're listed right here: the Pueblo Relief Fund, the Hopi Relief Fund, the Navajo and Hopi Families COVID Relief Fund, and the official Navajo Nation COVID-19 Relief Fund. If you can find in yourself to support these groups, they're really providing crucial assistance in Indian country right now. Little intro to Zoom. If there are any of you who have, I don't know, managed to be off of a computer for the last 7 months, well, welcome. This is Zoom, and there's a couple of tips that I have for you. One, you can move the talking heads. You might see Jonathan's and mine hovering, and we might be obscuring part of your screen. Just click the black bar at the top of it, and you can drag us around. Or if you click the little rectangle in there, it minimizes our heads, and we'll be out of your way. We'll be taking questions throughout Jonathan's lecture today. If you have a question that is of a particular technical capacity, he said something that uses some big word like stable isotope analysis and then just moves on, and you're like, what is that, Jonathan? Ask it in the Q&A button, which is at the bottom of your screen. If you're on mobile, click your screen and you'll see the Q&A button show up. I'll ask that right away. Otherwise, I will hold your questions to the end, and we'll have a good question and answer discussion with Jonathan at the end. If you're having difficulties, you can head over to our live stream on Facebook. You can get there fast by going crowcanyon.org/Facebook. And if all else fails, you can catch the recording of this lecture. It'll be posted overnight, and that'll be at crowcanyon.org/YouTube. Our mission at Crow Canyon is to empower present and future generations by making the human past accessible and relevant through archaeological research, experiential education, and American Indian knowledge. And our speaker today is really the product and one of our best spokespeople for Crow Canyon's mission. Jonathan has been an intern at Crow Canyon. He's been a research associate. He's been a scholar and educator for our college field school that we do in the summer. And his research really speaks to the public, speaks to ecological resilience, speaks to environmental revitalization in the Southwest. Jonathan Dombrosky is a PhD candidate and the Hibben postdoctoral research fellow at the University of New Mexico. And he's a research associate at Crow Canyon. He received both his bachelor's and master's degrees at the University of North Texas. His dissertation work, which is funded by an NSF doctoral dissertation improvement grant, focuses on better contextualizing ancestral Pueblo fishing strategies on the middle Rio Grande region of New Mexico around the Pueblo IV period. He is generally fascinated by southwestern zooarchaeology during the late prehispanic and early historic periods, and he leads projects centered on understanding raptor management and the intricacies of garden hunting, which we'll hear about today. He has broad interests in human behavioral ecology, stable isotope ecology, conservation biology, and environmental science. And his research has been published in international journals, including Archaeological and Anthropological Sciences, the Holocene, Hydrobiologia, and Peer J. And I skipped one slide. I'm sorry, Jonathan. I got to go back. We've got one more, our last webinar of the season. We're not meeting next week. Next week's Thanksgiving. A week later, on December 3rd, at this time, we'll have Dr. Todd Suravel presenting on the Barger Gulch site, a Folsom campsite. So that'll be really exciting. And our last one until January, we'll kick back up the first week of January. So without further ado, I want to introduce our speaker today, Jonathan Dombrosky. I'm going to stop sharing. Jonathan, you can share your screen now.
Sounds great.
Mm-hmm.
Here we go. Bring this up. Can you see it?
Looks great.
Awesome. All right, I'm going to get going. Thank you so much, Kyle, for that lovely introduction. And thank you so much for to Crow Canyon for inviting me to give this talk. You know, missing doing college field school, I've been doing it for about 4 or 5 consecutive years, and missing it this summer really threw me off, and I really miss everyone over there. So it's great to get to talk to everyone. And this kind of, the kernel of this talk, actually, I gave at Crow Canyon last summer, and now I've kind of expanded on it a little bit. And so before I launch into all the details about this, I just want to say that this is actually a collaborative work at the UNM Zooarchaeology Lab with co-authors. My co-authors are Caitlin Ainsworth, Abby Judkins, Yana Meyer, and my advisor and mentor, Emily Jones. So it's not just me, I'm just talking about it today. And so really the idea behind this talk is to kind of pick up on a pattern that I've noticed after doing, I've just started thinking about this the other day. I've been doing Southwest Zooarchaeology now for like almost, I think we're going on 10 years. So it's my decennial anniversary. And it's this pattern that I've picked up about garden hunting. This kind of when I talk to Southwestern zooarchaeologists, and just to define garden hunting really quick, we'll get into the definitions of it. Garden hunting is simply the acquisition of animals, the hunting of animals from agricultural plots or gardens, okay? And what I've noticed talking and doing Southwest zooarchaeology is that we just assume that garden hunting happened in the past. And sometimes it is our null hypothesis. It's just what we work with. Certain animals, especially rabbits, they're probably the ultimate example. Rabbits were garden hunted. And specifically, when I talk about rabbits, I just kind of want to define some terms. I might throw out these fancy terms. I might say leporid or lagomorph. And all I'm talking about, that's a collective term for jackrabbits and cottontails, okay? And it's especially cottontails that we just assume that were garden hunted. And I'll get into the details about that. And so one of the things, though, that I kind of want to talk about before I really launch into stuff is my goal today is to get you to care about where bunnies were hunted in the past. Okay, where and in terms of garden hunting, and exactly were they in open environments, closed environments, were they in gardens or not? And it might sound pedantic. It might sound like just a little thing. Who really cares about this? But picking up on this pattern and understanding if garden hunting happened or not matters a lot. And we're going to get into it. I'm going to get into why this matters, some big reasons in the Southwest at the beginning of the talk. And then at the end, I'm really going to hit some of these points home. So let's get started. So garden hunting as a concept was first introduced in the mid to late 1970s by Olga Linares, okay? And it was a concept designed and picked up on in the American tropics, okay? And it came to be so influential and how we were thinking, because it was a whole new subsistence strategy, a specific subsistence strategy, right? This acquisition of animals from gardens, specific subsistence strategy that could have substituted for domestication, animal domestication, in the past. Okay. And so that's not a little thing, that's a big thing. And then it kind of got picked up in the literature and Southwest zooarchaeology, and we've talked about it ever since. But one of the other things, the thing that we're going to get back to, that's imminently testable about this garden hunting hypothesis, if this happened in the past or not, is basically that animals were drawn into gardens and agricultural plots because they basically, those agricultural plots provided food, those cultivated crops, and habitat cover. Okay, so we're going to talk about that a little bit more. But first, I want to talk about why it kind of gained so much traction in the Southwest, which it has. And the reason, it's explained a whole bunch. It's helped explain and frame some patterns that we've picked up. One of those patterns is that in the P4 period, the Pueblo 4 period, especially around where I live, around Albuquerque, and the Pueblo 4 period is between roughly 1300 and 1600. We've noticed this pattern, zooarchaeologists, people who study animal remains from archaeological sites, we've noticed this pattern, that there's actually a lot more birds in these that get excavated from sites, a lot more bird remains, and they're more diverse. We can talk, we can talk about this pattern a little bit more, but that's generally what people think. And I actually had the chance to go to Bosque del Apache, which is just it's a wildlife refuge that's about an hour south of Albuquerque. And this weekend, I took this picture. I got to take a look at the sandhill cranes that are there, and I also got to take a look at the snow geese that are there. And so, anyway, getting back to the archaeological pattern of garden hunting. The idea has been that these fields that were laying fallow, right, along the Rio Grande, actually drew in all of these birds, and that ancestral Pueblo people were able to hunt these birds for their feathers or for meat. And garden hunting is the reason this all happened. This uptick happened in bird remains, and it's actually worth noting at Bosque del Apache, they actually they plant corn and wheat to draw birds in and feed them. These like big populations, so people come and kind of check them out, which is interesting. So garden hunting explains all of these birds. It also has helped. This is a schematic of McPhee Village. That's one of the first major villages in kind of the ancestral Pueblo world, and garden hunting was really kind of introduced into Southwest archaeology to explain how people were getting food in terms of early village life, which is pretty cool. But as a zooarchaeologist, what I'm really interested in, as Kyle mentioned, that I'm really interested in behavioral ecology, is how are people thinking about getting food on the landscape? Okay, and so what I'm going to kind of do, people who've seen the kernel of this talk have seen me do this before, but this is actually a picture of the gardens, the experimental gardens at Crow Canyon. You can see Crow Canyon in the background here. So what I'm going to kind of ask you to do is imagine you're a farmer, and you're tilling and you're cleaning, maintaining fields, and you can kind of see this deer in the background there. I don't know, I just highlighted it. I'll highlight it here. There's deers back here, and there are 3 bunnies, right? And you need some food, you need some meat, you need some protein. So what is the logic that you're going to use? Are you going to go further afield and go get that deer, or are you just going to take the bunnies that are already pests in the field, right? And so you might bop these bunnies on the head and you might take them. And this is garden hunting in a nutshell. Okay, are you using your agricultural fields, your gardens, as a protein trap? That's garden hunting. Okay, and if you could maybe use different technology to maybe, I don't know, like a net to scoop all these bunnies up at once, you would definitely start garden hunting. So garden hunting is important when we start thinking about the logic of what foods are being selected in the past and why. And what zooarchaeologists like calling this is diet breadth, right? So what are the animals that are the highest ranked in the diet of people through time, and how does that change? That's a question that we're all really interested in. But depending on how people are making decisions about getting food, is it based off of how far things are away? Are people garden hunting? Rank can switch through time. Okay, so this is actually how I really got interested in garden hunting and the assumptions that we make about how people are getting bunnies over deer or turkeys or not. I hope that makes sense. Another reason that actually garden hunting has become is almost transformed through time is thinking of what archaeologists term niche construction theory. And niche construction theory is really just interested in how humans modify their environments, right? And modify the environments of other animals to impact themselves, to impact humans and those animals, and how we modify our own niche, our own place in the environment. This is just a picture of El Cerro de Trincheras, which is a terraced, all these terraced hills, which are beautiful. Garden hunting really hasn't. I don't know much about garden hunting in this context, but in terms of niche construction, we're kind of focusing on how we're like domesticating ourselves and how we can change our own foraging logic. So anyway, it's just changed through time a little bit. The other thing that's kind of when we think about garden hunting that a lot of people say is that it doesn't necessarily have to be a an intentional thing. It can be opportunistic. Okay, it can be indirect. People don't necessarily, and where I'm kind of going to argue, bump up against this idea. People don't necessarily have to think of their fields like protein traps. They can just be doing pest management and getting a little protein, getting some rabbits, and kind of hitting two birds with one stone. But one of the things that I've started kind of doing, I've kind of been declaring war or started declaring war on this word in zooarchaeology, opportunistic. What does that mean? It's really vague. And I would argue there is a big difference between wanting an opportunity to arise, like bopping a bunny on the head in a garden, or not wanting that opportunity to arise. And that makes all the difference in the world. If what we see in the archaeological record, in the zooarchaeological record, the majority of the signal from the bunny remains that we see, if those were garden hunted or not, okay, it matters for a whole slew of reasons, and I'll get more into those a little later. So the other thing that kind of matters when we think about garden hunting itself, if it's a viable strategy or not, one that people are using a lot or not, is we have to be willing to say that maybe it didn't happen at all, right? Because the idea with gardens is that you don't really want pests in the garden in the first place, right? So taking care of the problem of the pest beforehand, and there's actually evidence of before crops are being planted, of in some areas where groups will go out and do communal hunts of rabbits, which also could help in this. So the idea here is maybe it wasn't so important through time, and maybe the majority of the signal that we see in the zooarchaeological record, they weren't garden hunted. So that's what I'm going to be talking about today and showing you some kind of data that I have on that topic. Getting back to this is a picture of a Dakota woman in the 1850s, kind of guarding her cornfields in this shelter, this stand, and she's banging this pot to scare crows away from the corn, this cornfield. And we know from Antonio de Espejo in the late 1580s that around the Rio Grande, that these same shelters and people were watching gardens to make sure that pests weren't eating them in the first place. Okay, so we have reason to believe, okay, that garden hunting, you didn't want to garden hunt, right? So I hope that makes sense to everyone. The other thing, and I got to thank Kyle. We kind of nerded out yesterday about this topic. He reminded me of a paper by Stephen Son-Jordet that talks about how pest management actually works in Hopi fields. This paper was published in 2017 in the Journal of Ethnobiology. And one of the things that Stephen Son-Jordet, we could talk a little more about this in the Q&A section. One of the things that Son-Jordet talks about is that in times of drought, pest management would become a little more intense, but pests become a more of a problem in agricultural fields, because actually the agricultural fields, the plants in them, have water, right? And it's those things that say rabbits would want. Okay. And the other really interesting thing that Sanjordet brings up in this article is that how people decide, where Hopi people decide to put fields. Are they either going to plant fields further afield, and what he calls these are extensive strategies, making more agricultural fields, or are you going to make fewer agricultural fields but watch them like a hawk and make sure they aren't predated upon? And so what basically Sanjordet says in this paper is there is a point at which making fields further afield during times of drought doesn't make any sense, because they're going to get predated upon. It's better to keep fields closer to home that you can maybe bring water to, maybe through irrigation or something like that. And you can make sure they're not getting predated upon. And we're going to kind of get back to this. And that's why I wanted to make sure to kind of talk about this. Okay. The normal story of garden hunting in the Southwest, I kind of want to highlight this for people that may, if this is still a little unclear for people. So the normal story of garden hunting through time in the Southwest, here on the y-axis, we have the reliance on garden hunting, and then we have agricultural intensification just in general, the amount of agriculture that's going on through time. The idea that we kind of think, which is kind of assumed in the Southwest, is that there's like this linear relationship, right? As agricultural intensification increases through time, we also get more of a reliance on garden hunting. So the idea here is human population must be dense. There's resource depression in terms of larger animals like deer. In the general area, populations are increasing with agricultural intensification. Humans become more restricted in terms of their mobility because they're more invested in agriculture. These farms, these fields create habitat for small game and food, and then it just becomes really inefficient for people to travel long distances. So this garden hunting becomes this strategy that is better, right? And so we assume that maybe, I think in everyone's kind of head, we just kind of have this idea of this linear relationship. And this is kind of what I was taught when I talked to Southwest folks. But maybe it's a little more, maybe it's not a linear relationship, right? Maybe it's a curvilinear relationship where there's just some inflection point where the amount of garden hunting just levels off through time. And maybe that inflection point occurs at some period of really intense agriculture or really intense agricultural intensification, like the Pueblo II period, which is around 900 to 1150. That's when a lot of agriculture is kind of going on in the Southwest. But maybe as well, that reliance on garden hunting actually has to do more with foraging efficiency. And when I say foraging efficiency, all I'm saying is the ease at which high-ranked prey, like deer, let's say, are procured and then consumed, right? And as foraging efficiency is, when you have lower foraging efficiency, maybe you're relying on garden hunting a little more, right? And when it's high over here on this graph, you're not relying on it a little bit more. Or maybe, right, there's a lot more that we can say with garden hunting and come up with hypotheses about it instead of just assuming that it happens like this. Maybe it's the ability of people to trade with other people for food, right? Maybe when that's super low trading efficiency, that you would rely more on garden hunting, right? It's way more dynamic than this to rely on this very specific strategy. In terms of foraging efficiency, which is what I think about a lot as a zooarchaeologist, maybe right here is what we're interested in. When foraging efficiency is really low, we would expect people to be using garden hunting more. This is what I would assume. Okay. So we're going to get kind of back to this when I kind of bring up a little bit more data. But I want to talk about, before that, how we normally think about measuring the presence or absence of animals that were garden hunted, right? And we normally do this kind of in an indirect way. We normally think that, we look for lots of species maybe that are that really prefer modified habitats and that are generalists, right? So cottontails and jackrabbits are generalists. And so if we see a lot of them in the zooarchaeological record, a lot of their bones, then maybe we could say, oh, that's evidence of garden hunting. And that's how it normally is done. The other way that's pretty common is to create a wild kind of baseline. What does just a normal baseline of animal bones in the wild look like during this particular time period that you're interested in? And then the thing that you recover from the archaeological record, is it different than that? And is it filled with those generalists that really like those modified habitats? So this is another way that this has been done in the Southwest. I hope this is all making sense. I see some questions coming in. Maybe I glossed over something. Maybe not. Okay.
Oh, you're good. You're good.
Okay.
Keep going.
Okay, cool. The other way is that this has been inferred in the zooarchaeological record is that garden hunting is viewed as a kind of a non-selective form of hunting, right? So it's just anything that is in that garden, people are going to be hunting. So if you have really diverse kinds of animals that are found, all these bones, maybe that'll be evidence for garden hunting. And each one of these kinds of ways of inferring garden hunting in the past is, like I said, indirect. So maybe, and what I'm going to talk about here in a little bit is a more direct way of maybe doing this. But one I want to talk about too, before I get into all of this, is that we think there are differences. I said I'm going to be talking about rabbits today, right? But there are slight differences between the jackrabbit, which is here on the right, and then the cottontail, the desert cottontail, here on the left. Okay. There are slight differences in the kind of habitats or micro habitats that cottontails prefer over jackrabbits. They have different predator avoidance strategies, right? So the cottontail prefers to hide in densely vegetated areas, whereas the jackrabbit likes open areas so it can run really fast and get away from a predator. And what we think actually in the zooarchaeological record in the Southwest is that people were hunting and butchering actually these animals in different ways, and it's really important to compare them, which I will do when we're thinking about garden hunting. And I want to give a shout out actually to a chapter that recently came out in this book. This book recently came out. Patty Crown recently published this. She edited a volume called The House of the Cylinder Jars about room 28 in Pueblo Bonito in Chaco Canyon. And some of my co-authors on this study that I'm talking about today, Caitlin Ainsworth and Emily Jones, and also Stephanie Franklin, was part of this one chapter where they looked at bones that came out of animal bones that came out of room 28. And they actually, let me turn on the laser pointer. They actually compared the kinds of bones, the types of bones that between jackrabbits and cottontails and the large trash mounds outside of Pueblo Bonito to the ones found in the subfloor of room 28. And they actually picked up on a difference where jackrabbits and cottontails seem to be butchered differently, right? It's probably based on how they're captured in the first place. Or maybe jackrabbits, and they do the same thing over here with really specific, this is cranial remains. Axial is just vertebra and things like that. And appendicular bones are just your appendages, the bones that make up your arms and legs. And so the idea here is maybe actually jackrabbits represent these communal hunts with maybe big nets, and people do them further afield, get all these jackrabbits, and maybe they butcher the really high, great, meaty parts of bones and bring them back. And maybe cottontails are found closer to home, and they're just brought whole, right? So we see these differences. And so I just want to talk about that a little bit. I'm going to get into this a little bit more with jackrabbits and cottontails. So we would expect to see differences maybe in where they're kind of residing. So are there more direct lines of evidence, okay, to test for garden hunting in the past? And if you remember, in that original paper by Olga Linares, she says, the reason why animals, garden hunted animals, are coming into those fields is because they offer food. And luckily, we can test that. We can test in archaeological remains using bones what animals or resource pools were animals relying on in the past to maybe more directly test this garden hunting hypothesis. I hope that makes sense. So the idea here is the stuff that's in the garden, was that what was being consumed by those animals? And this is a more direct approach. So the whole thing that I'm presenting today is, do rabbits look like they're eating things that humans are eating? That's the idea. And how I'm going to do that is with stable isotope analysis. I'm going to get into the details of that right now. So anyone at Crow Canyon who's seen me talk about stable isotopes has probably seen this graph. Stable isotopes, I'm going to keep this really general, okay? And if people have questions about this, they can bring it up and I can get into all the little details about this if they want. But I just kind of want to teach you really quick how to read a stable isotope biplot, kind of what it means, and then we'll get into kind of some data. So we've got two axes here. The x-axis is what we call delta carbon 13. It's just carbon. What we're looking at is of a specific tissue type of an animal, or say you, if I tested it, maybe your hair or a fingernail, what weight of carbon, okay, which is an isotope, makes up the proportion in you, okay? What's really cool is that different plants that do photosynthesis differently, okay, separate out in this delta carbon 13 space. The majority of biomass, we call this C3 photosynthesis, don't have to really get into the details of it, on the globe does C3 photosynthesis, and they appear towards this end of this axis right here, around, and we call this fancy looking percentage thing right here, we call this per mil, not percent, per mil, right? So C3 plants, say like a cottonwood, okay, occurs over here around negative 26 to 28 per mil. Whereas plants, more arid adapted plants, like say, I don't know, blue grama grass or corn, actually corn does this, does photosynthesis differently, and it separates and is more towards this side of this x-axis, okay? So C4 plants, we call them C4 plants, that's the type of photosynthesis it does. It's around negative 12 to negative 14 per mil. Okay, so that's how these sort out. So again, if you were eating a lot of corn in your diet, which most of us are, and I took a piece of your hair, wherever, and I could plot it in this space, it would likely be over here. Okay, and that's what I'm going to do with animal bones, and I'm going to kind of build graphs build these little areas in front of your eyes. But now we need to talk about the other axis right here. We call this delta nitrogen 15. And all this does is it basically is a measure of where you're at on the food chain, or where an organism is at on the food chain. Okay, so say a primary consumer like the cedar waxwing, I really like birds. Cedar waxwing is down here, whereas say a golden eagle, a more tertiary consumer, will be up here. So to kind of break this down again, say you're over here. So if I took a piece of your hair and plotted it down here, and you occurred right here, the little dot right here, I would infer that to say you were eating more C4 plants and you were lower on the food chain, in the food web. We call that trophic positioning. Okay, I hope that makes sense. The only way basically the whole way to do this is just to reconstruct what resource pools animals were relying on. I hope that makes sense to everyone. Okay, there's only one study that I know of that has used isotopes on rodents and on lagomorphs, jackrabbits and cottontails, in the Southwest that has used isotopes and has inferred, okay, garden hunting happened in the past. And so what we see here is just basically a histogram. This is the number of samples. This is from Deanna Grimstead and her colleagues in a 2016 paper. This was not the main point of their paper. It was more of kind of an ancillary point that they made. And they basically said, hey, we picked up on garden hunting with stable isotopes. And what you can see here is they compared from modern non-agricultural animals from museum collections, their bones, and they compared it to modern animals that were recovered from agricultural fields. So in the yellow here, this is where they kind of distribute in carbon space. I'm going to be using that term a lot, carbon or nitrogen or just isotope space. This is where the majority of them are. And then the modern agricultural ones, like rabbits that were taken from fields, are over here in gray. And so they basically asked, okay, where do our fauna, our rodents and our jackrabbits and cottontails from Chaco Canyon, this is where these came from, where did they plot out? And they basically said, oh, they overlap with these modern agricultural ones. These were garden hunted, right? So all these blue ones are those archaeological animals. So this is evidence of garden hunting. And so one of the things that I have recently published on is actually a correction with carbon values. I'm not going to get into the details of it, but if we apply the correction to Deanna Grimsted's to her data, things become a little less clear about kind of this bridge between this kind of garden hunting signal that they basically said. I'm going to go back a slide. Basically, there's a buffer right here, right, where they said, okay, this is basically what the idea was, this is where garden hunting is happening. But if we apply this correction, we can see actually that buffer disappears. This isn't necessarily surprising because, and we can basically say, actually, it's just kind of this continuous pattern that we see, and separating garden hunted stuff from not garden hunted stuff is a little more difficult. I hope that makes sense. And this is for a very particular reason. This doesn't necessarily say that Deanna Grimsted is wrong. It just makes it a little less straightforward. And the reason it is less straightforward is kind of something that I really want to, one of the main points that I want to hit home today about people using isotopes in archaeology is that delta carbon 13, right, of something that is around, of a value that is around -12 to -14 per mil, right? C4 plants, that's what we said, that do photosynthesis in that way, arid adapted plants. It doesn't mean corn, okay? That's not the point, all right? It's not a taxon. It's not a type of species of plant that you're identifying with these values. You're identifying a photosynthetic pathway, okay? Or it could be grass that she picked up on in that C4 grass that made its way through the food web, that channeled its way through the food web that she picked up on there. And that's because the Southwest has a lot of arid adapted plants just naturally occurring, okay? I'm going to run us through really quick a case study that people did in the panhandle of Texas, okay, where they compared bunnies, what bunnies, desert cottontails, ate, and agricultural little playa basins, little divots, ephemeral lakes, you could call them, these little ephemeral lakes that are found on agricultural property versus not agricultural property. And they wanted to see, what were these bunnies eating? How were they shifting their eating preferences? And I went through, you could see all the animals, they looked at their gut contents. And I went through and calculated, okay, from those agricultural playa basins or just the normal playa basins, what is the majority, what is the proportion of C3 and C4 plants in their diets? This was all during summer, by the way. There's a seasonal component to this. The number one ranked thing in their diets, no matter which context you look at, is C4 plants. And in just normal playa basins, C4 plants are way more abundant in the diet. This is because desert cottontails and jackrabbits are generalists, right? They're going to eat what's around, and what's around is C4 plants. So we need kind of a better way to directly test garden hunting. We can't just do it based on the values that we get when we do stable isotope analysis. I hope this makes sense to everyone. It's a really critical point. What we actually need to do is we need to compare these animals to human isotope space. Okay, where do humans occur in their position in this bivariate plot? And do these bunnies start kind of converging in that same space? I call this niche space. You can just think it isotopic niche space. It's a jargony word that I use. It just means the area. Okay, so what I'm going to do right now is I'm going to walk you through. I'm going to build a graph before your eyes and define space where different kind of resource types occur, where humans occur that we know from in the past, and then from a couple of sites, which I'll get into, where bunnies occur. So this is where beans are, right? So right now I'm kind of defining, what does the isotopic garden look like? Beans are right here. Squash is right here as well. This circle, this ellipse, is a 95% confidence ellipse. So we can be 95% sure that the data that I have here occur in this space. So I basically tested all of these from seeds, from native seed search. There are heirloom varieties of seeds that I analyzed. So there I have 10 beans. I have 8 squash seeds, and then I have 20 corn seeds that I analyzed for this. So this is where this all occurs, in nitrogen and carbon isotope space. So now, and we can be 95% confident about that. And now we can say, okay, where do humans occur? Okay, and so these are human remains that were sampled for isotopes, and they are from multiple different contexts, all the way from Basket Maker 2 until Pueblo 3 that people had analyzed in the past historically. And so I kind of went through the literature and compiled all of this of what we know to say, okay, broadly speaking, what is human isotope niche space? How can we define that? This is 73 specimens that we have here. Okay, I hope this all makes sense. And what you'll notice is that they overlap, especially in carbon space, almost 100% in carbon space, with corn, right? People are eating corn. That's the idea. Okay, so now where are other animals at? And there's a huge literature out there on turkeys, and that's all this means right here is Meleagris. That is the genus for turkeys, Meleagris gaiapavo. And people are really interested in when did turkey domestication take place, and how can we tell that with stable isotope remains, or stable isotopes on turkey remains. And the idea here, right, is that these C3, these more C3, I should say, turkeys, right, they're clearly separate from these turkeys over here. So there's like, I don't know, 23 turkeys over here from a huge database that I collected. And then there's like 180 turkeys over here that completely 100% are encompassed. Their isotope niche space is encompassed by humans. And so this overlap has been used to say, okay, these are domesticated turkeys. I'm arguing we also need to infer garden hunting in the past. We need to use this same approach. We can't just say, oh, they occur somewhere where corn should be, therefore it's corn, because we can't rely on that, because C4 plants naturally occur in the desert Southwest. Okay, so I was interested to go to a couple. I have two archaeological sites that I'm going to talk about today. When I started this project, Hummingbird Pueblo is one. It's a large site. It's right here. A large Pueblo 4 archaeological site. It is Pueblo 4 basically is, like I said it before, 1300 to 1600. The idea here, the logic why I wanted to look at bunnies from these contexts is that this is when we think agricultural intensification is very, very high. Okay, because populations are really high. These are some of the largest, in terms of room blocks, some of the largest pueblos that we see in the Southwest, and Hummingbird is a very big one. So the idea here, the logic is intensive agriculture, right? People aren't going to really want to go very far away, and then maybe garden hunting is a really advantageous subsistence method to use. And so I took about 17 jackrabbits and about 19 cottontails, right? Silvolagus right here is cottontails, and lepus is jackrabbits, okay? And I sampled them, and I said, okay, how much do they overlap in isotope space with humans? And the answer is, as you can see, not a crazy amount, not a significant amount. They overlap a little bit. I think it's about 5.5% of the jackrabbit niche space overlaps with humans, and it's about 4.5% for the cottontail niche space overlaps with humans. So maybe some are in a garden, but not a substantial amount. Not enough to assume that garden hunting was happening all the time at Hummingbird Pueblo. Maybe a little bit, maybe. We can get into some details about this. I hope that makes sense too. The one that I'm really excited today to talk about is Sand Canyon. This is brand new jackrabbit and cottontail data that I'm presenting on here. So I was lucky enough to get permission to sample jackrabbits and cottontails from Sand Canyon Pueblo. And so this is this Sand Canyon, if people aren't aware, is a really magnificent site that is in the Mesa Verde area. It is occupied right up until the last bit of major Pueblo occupation in the Southwest, which is around the Pueblo, what we call the Pueblo 3 time period. The 1300s is when basically people leave the Mesa Verde area. And what we know, what's really interesting, the logic that I had for wanting to look at rabbit remains from this area is we know that foraging efficiency is really low. There are really no artiodactyls, or sorry, deer. Deer are really rare. They're really small parts of these assemblages, right? So the reliance on basically lagomorphs and turkeys increases through time. We think they're really important during this time period, right? And right at the last breath of occupation, we actually think cottontails are really important in the diet of Pueblo people. And this has been basically attributed to, this is where the assumption really comes from, is that garden trapping is the reason that cottontails are so important during this time period. And so I wanted to ask the question, were those cottontails eating garden stuff? And I can tell you, they really weren't. There is not a lot of overlap at all between jackrabbits and cottontails and humans. Okay, I've got about 14 jackrabbits here and about 16 cottontails that I can define the space for. So it seems at Sand Canyon, these lagomorphs were not in the garden. They were not being garden hunted, at least from this sample. Okay, I hope that all makes sense. Okay, and I want to give a shout out too. This paper literally got published 2 days ago. It's on the first view. This is in the journal American Antiquity, the flagship journal for North American archaeology. Eric Gary et al. And this is a really cool paper where they basically talk about using isotopes, how you can think about resource depression, right? So basically people maybe overhunting things, overhunting animals, and how we could pick up on that archaeologically speaking or not. And so I'm kind of going to paraphrase for them. I don't know if they would say it like this, but they basically talk about in this paper, people who are interested in this kind of stuff, please go read this paper. It's cool. They suggest this parabolic relationship between a garden hunting isotope signal, no matter how you would want to define it. I define it with overlap with humans, and harvest pressure, how hard people are hunting certain animal types. So rabbits in, say, a garden patch, right? They would basically say you would have a really low signal of garden hunting from isotopes if rabbits weren't allowed at all in the garden patch. And then it would only increase if they were allowed in. But at some point, you're going to start overhunting or depressing those populations of rabbits. And they're just not going to be able to make it into a patch or a garden patch at all because they're just not around anymore, right? So resource depression is occurring. So that brings up a really important, kind of question is, maybe it's just that rabbits aren't around at all. Cottontails aren't around at all locally around, say, Sand Canyon. And that's why we're not seeing this signal of garden hunting. Maybe their populations are getting depressed. But that's not really what we see, okay? In terms of, this is work done by Robert Muir and John Driver in 2002. And what you can see is we have percent of the kinds of collections that include bones, and this thing called the lagomorph index. And you can just think about that as the amount of cottontails that are in collections, okay? Is it a lot or is it a little bit? And basically, this bin right here, 0.81 in terms of this index to 1, is like basically totally dominated by cottontails. And what you see during the Pueblo III time period where Sand Canyon occurs is that there are a lot of cottontails that are around. These populations, there is some evidence of resource depression that's starting to happen on the populations, but they're not gone from the landscape. They're around. They're quite abundant. So this leads me to conclude that it's not resource depression, okay? That these bunnies were being excluded from the garden patch, okay? And this makes sense when we think, going back to that one paper by Stevenson Jordet. What he basically said was, if during times of drought, which we think the terminal Pueblo III time period is, maybe it would make sense to bring garden patches closer to home and to really watch a very selective few garden patches, right? To be more intensive with your gardening practice than extensive. And maybe that's what we're picking up on in Sand Canyon. Maybe we can talk a little bit more about that. I've got about maybe 3 more minutes to wrap up here. I don't want to keep people. One of the main reasons why garden hunting really matters is in the domestication literature, we're getting to this point, right, where we are starting to talk about how did animals get on the domestication pathway? And managing food for animals is a major way that this happens. And already in Mesoamerica, this is one paper that was published by Andrew Somerville et al. And PLOS ONE, a really cool journal, that they argue actually rabbits, cottontails and jackrabbits, were being domesticated. They were not on the pathway. They were domesticated in Mesoamerica. And basically what I want to say is this really matters for domestication studies because, man, we got to be careful if garden hunting's happening in the Southwest or not, because we're going to start getting into this kind of literature, which is a big, domestication studies are huge, right, in anthropology and archaeology. So there are some take-home points that I kind of want everyone to walk away with. I'm not saying garden hunting wasn't a thing in the past, in the Pueblo past. Okay, we know it happened. It happened. But how much of it happened and how prevalent was it? We need to know this. It is not something that we can just assume that happened. Garden hunting matters. Okay, it matters for understanding foraging logic, for how people are basically using different parts of the landscape to understand traditional ecological knowledge. How are people understanding food security and mitigating against food insecurity in the past? And how are they using different subsistence strategies to do that? Food security is really important today. And how does that change through time and under what environmental contexts? It's really important. Okay, garden hunting is not a trivial thing. It also it's important because we need to understand how far people are traveling on the landscape. We need to understand how people are interacting with the landscape. We need to understand how during these time periods, different animals are interacting on the landscape. We need to understand farming practices. I just talked about domestication studies, right? So garden hunting plays into all of these different anthropological and archaeological topics. And the last thing that I want to make a point with people interested in stable isotopes, C4 does not equal corn. It is a photosynthetic pathway. We need to understand interactions with humans, right? That's how we're going to start more seriously thinking about garden hunting. I hope that made sense. So these are some of my acknowledgments. My Twitter handle is right up here, right here. I really want to thank, again, Caitlin Answorth, Abby Judkins, Yana Meyer, and Emily Jones. Thank you, Taylor and Kyle, and thank you to all of these other people. I'm going to stop sharing my screen now.
Hey, thank you so much. That was great.
That was fast.
It was fast. I know. It's so hard, but no, it was really good, and people are really excited about what you were presenting. So
can I actually interrupt? I forgot to talk about something.
Uh-oh.
One of the is that okay? Just to say something really quick.
Oh yeah, no, do it.
The idea with people around the basically guarding garden patches, there's this idea in ecology called the landscape of fear. Okay, basically, if people are around and guarding garden patches all the time, bunnies aren't stupid. Okay, we assume this, that they would just wander into fields, when they're being predated upon constantly, right? They do kind of a risk management, a predation management strategies, how they move around the landscape. And so the risk during times of drought, which is really cool about that Sun Jordet paper, it seems like they're kind of doing some risk analysis and going, I need water. I'm going to go into this field. I know I could get shot with a shotgun, but yeah. And so to think that this didn't happen in the past, of course this happened in the past. So,
got it.
Yeah.
So I want to start with there's a lot of questions coming in, and people keep them coming in. This is great. I want to start with just I need a clarification from you. So, and it's about Grimsted's analysis.
Yeah.
I thought the logic of that analysis was that wild animals of the same species in contemporary environments where agriculture really isn't happening don't seem to be eating a lot of C4 plants, whereas the archaeological animals and specimens that were taken from agricultural contexts were eating a lot of C4 plants. And you start.
Oh, you went mute. Or is that maybe me? No, that's not me.
Uh-oh.
Oh, you're back.
Okay, good. You started talking about playas in North Texas and that sort of thing, but can you just walk me through that one more time? Why those data of showing wild animals not eating a lot of C4 plants, these archaeological animals apparently are.
Yeah.
What's the logic there?
The logic there is basically C4 plants aren't just, right? They aren't just in fields. They aren't in gardens. They're around the natural habitat. And there's a environmental component to that too. During more arid time periods, maybe you would expect C4 biomass to increase, which is the idea with looking at the summer, right? There's a seasonal and climate kind of thing that we need to think about there. So maybe it's not that they were in gardens. Maybe it's that they were just out in the field, or they were just out in the open habitat. Do you see what I'm saying? It's not.
No.
Maybe I'm not understanding.
No. The left. I thought the left side of that graph, and maybe I just wasn't reading the graph right, but I thought the left side of the graph was animals that are out in the environment.
They are
in that same environment,
right?
And they're apparently not eating that many C4 plants.
So
your argument is that there are a lot of C4 plants that are around.
Yes. And so, but saying those ones that trailed off to that more C4 area aren't wild that's an assumption. They could be wild. They can. There's equifinality there. So in terms of why I say this, why I say that she's not necessarily wrong is because she actually has the abundance next to it, right? They're more prevalent. But saying where that cutoff is where things get confusing. And so we can't. I don't think relying on the values themselves are going to tell us much, because we could go, well, those could just be wild. Just because we have a bunny bone that looks like it was C4, that doesn't mean corn, or an agricultural field. That means C4, right? Which occurs naturally out here in the Southwest. And so, again, not saying she's necessarily wrong, but I think my argument is the better way to do that is overlap with humans. Because they're the ones who are assimilating the garden, right? And they're the ones who we need to compare to with those animals.
The comparison at Sand Canyon was real compelling.
Yeah.
Okay, here's a challenge. In a minute, maybe a minute 30 seconds, explain how stable isotope analysis is done.
Okay. Oh, how it's done. Oh,
how it's done. People want to know, what is the process exactly for finding the isotope levels in beans, corn, or humans? But you only have a minute.
Okay, well, I'll talk about it with bone. It's different for different tissue types. It's different for different animals and things like that. For bone, what you do is I'm interested in collagen. You can do different things. So what I do is I basically dissolve bone in a weak acid, and then it becomes just flexible jelly collagen. I dry that down, I weigh it out, and then I put it in a little capsule, and I actually hand it off to people who work with the instrument. But a stable isotope, or a isotope ratio mass spectrometer, which is the instrument that's used, takes that little capsule. You essentially set it on fire, you set it ablaze, right? So all of its elements are particleized. And then you put them through a mass spectrometer, which just measures the weight of different chemical elements. We can get into, if people want to know how that works, but you just count the number of those different heavy or light elements there are for carbon. And then that per mil number that I gave you, is a ratio of light stuff to heavy, or heavy stuff to light stuff, if that makes sense.
Yes.
And so how the photosynthesis works is a whole nother thing, and how plants do that. It's amazing, okay, that plants were able to evolve and use different weights of chemical elements to do things a little bit better. It's pretty mind-blowing, actually. So anyway.
And it's also amazing how little material you can get. Isotope.
Oh, yeah. It's very little.
Yeah.
It's milligrams.
There was a great question that came in, and it really piqued my interest, too, because I don't know the answer to it. Dogs.
Oh.
So I mean, you talked about turkeys. Talk about dogs in two ways. Way number one is dog isotopes themselves. What are they eating as another human domesticate? Dog question number two is, did Ancestral Pueblo people use dogs to help guard their fields or hunt rabbits? Do we have any evidence of that?
There is ethnographic evidence of that. I was just reading about that the other day, that dogs were used, and I got really interested in that. I would be remiss if I didn't point the audience who are interested in dogs to Victoria Bowler's work, who works with my advisor and mentor, Emily Jones. She is doing her dissertation, NSF-funded dissertation, on dog remains and dogs in the Southwest, and has done isotope work. So this information is out there. But just to go really fast over this, it's the same idea, is overlap with humans. And there has been a number of studies on the West Coast in California that have done this, that have been really great, that have basically looked at the convergence with humans in isotope space. And the idea is they're eating the same things, right? bringing dogs into the domesticated into the house, right? They're going to overlap in niche space with humans. And that's the important thing. It's been used in the same way. And garden hunting is the same thing. That's my argument, at least.
Got it. Do you get evidence in bunnies and hares that are found in human trash piles, so in middens, of them also being and they appear cooked or whatever, of them also being gnawed by carnivores at all?
I've seen it. I've seen punctures
You've
on
seen it?
carnivore. Yeah. But that's refuse. I would think if I would actually, man, there's so much.
I
could go down this
a
for
bunny
a while. Right. But it's probably, just they're chewing on stuff around the trash deposits. That's the idea, right? With dogs, they're just around getting what they can.
Right.
Yeah.
Yeah. You've got some good questions from some people you'll know. Joan asked, logic says to me that if I was a bunny and there was a field of domesticated, juicy, delicious corn nearby, I'd do my best to get in there and get some of that corn at risk of being captured or killed by the farmer. I'd choose that corn over other C4 plants I can get anytime. Is that not right?
It depends. So that's my entire argument, right? So during times of drought, maybe if we saw a garden hunting signature, right? Maybe that would actually, in the archaeological record, so when and where this is happening needs to be what we're talking about, not that it just happened. So if it did happen during times of drought to get water, that's a different thing. And there's isotopes we could use to test this. But
yeah, maybe. Yeah, there is.
Yes, there's a whole thing. I don't want to get too far down this. But the idea is, sure, it might be worth the risk, right? This risk. But if you can get enough food, when during times of not so much drought, there's actually a figure that I wanted to show. I won't show it. It's too complicated. From that Sun Jordat paper. Go check out that paper. It's awesome. It's not worth the predation risk. Bunnies aren't going to go into those plots, right? So we can't just think about bunnies as only being food oriented. They're also, hey, I don't want to die oriented too, right? And you're creating this landscape if people are watching these fields to make sure they're not being managed, right? So sure, what I'm saying is, who's the one who asked that question? You said I knew them?
John Goldstein.
Okay, the idea here is not. I'm not trying to categorically say garden hunting didn't happen, but it is far more dynamic of a process, and we can't just assume that it happened.
Well, and one of the implications of that Sun Jordat's paper, as I remember it, was just that rabbits and hares, lagomorphs, can do a lot of damage to young corn. Yep. And so the Hopi farmers that he was working with really worked hard to make sure that those animals weren't destroying their crops.
Yeah, they said 24 hours sometimes they were out there.
They might not have had time to eat that C4 plant and have it certainly have it enter into their bone collagen. Yep. I don't know what the collagen replacement rate in leopards is, but
So the idea, one of the things too, is that I'm just not aware of a scenario. Okay, when thinking of this replacement thing, collagen, it's a while. It could be like 6 months. But I'm not aware of a scenario where you would have the majority of cottontails from, say, Sand Canyon, right, that were garden hunted, and you would not be allowing them to not eat in those garden patches. Does that make sense? If you were getting them immediately every time they came into the garden, man, those bunnies, I'm telling you, those bunnies would go, I'm not going to die. I'm not going there. It wouldn't be the majority of the signal. You'd have to allow a baseline level of, say, predation on those crops to allow them to be the majority of the signal. So again, I'm not saying this didn't happen, but to be the majority of the signal, that landscape of fear thing, I think, is really important in this. The other thing that I wanted to note too is, you notice there was a lot of overlap between jackrabbits and cottontails in isotope space, right?
Yeah.
So they're eating on very similar things. So if jackrabbits aren't going into the fields, right? Those aren't the garden hunted ones. Then, but cottontails are garden hunted. Like it makes no sense. The overlap between those two matters. Their ecology is different. Their predator evasion tactics are different, but their feeding ecology maybe it's more similar. It is more similar. We showed it.
Got it. Here's an interesting. human behavioral ecology question. How does the protein or calories, yeah, you get excited when caloric value of bunnies how are they compared to something like a turkey, and how many bunnies would be required to be worth chasing and catching and eating?
I have no idea.
Well, no, but just generally go through again with us that sort of prey choice model, that relationship between the caloric or protein value of a prey and the decisions that an optimal forager would make.
People are going to say I sidestepped this question because I'm going to basically say something that's like totally dissatisfying.
Okay.
I belong to a school of zooarchaeological thought that says doing all of this kilocalorie stuff is stupid. Okay.
Okay.
And so the idea here, the question is not stupid in terms of what's the rank. That's important.
Yeah.
But so we do it based on body size, right? Those correlate with one another. So just in terms of things are all equal, it goes, and we're just foragers with, our hands or with, I don't know, a spear, which is not these contexts. Then the rank would go in terms of body size: deer, turkeys, wild turkeys, cottontails, right? That's the body size rank. So deer number one. But if I have a net, right, and I can get a massive amount of cottontails or jackrabbits in one fell swoop, the energy maximization kind of logic depends on all of that stuff. So that's why garden hunting matters. It also matters with how far am I going afield? How much energy do I have to expend to go get that rabbit? If I could just go to my garden plot right outside of my house, then the cottontails are more efficient. It's not necessarily about the calories. It's about it is about the calories, but it's the number of things that you can get too. It's not the calories in one package. That's what I meant to say.
Got it. So it's a triangulation between value, the presence. So I can't remember what they call it in the HB literature, but it's how often you come across that animal, and then how difficult it is to capture that animal.
Encounter rate and handling rate.
Oh boy,
that's all this stuff.
All right, good.
I hope that answered. That's a good question. But I answered in a different way. Body size.
Got it. So I actually had a question about this too, and it bears on the way that you talk about this in your eventual diss or a paper about this. You compare, when you do isotope space, you're comparing both nitrogen and carbon isotopes,
right.
And I know that turkeys eat some insects, and so that would raise their nitrogen, their trophic level, right? Yep. Do rabbits and hares why would you expect their trophic? I mean, not why would you expect? Why does it appear that their trophic level is high? Like, why is there a spread?
Right. So in terms of them eating insects, I'm sure you could find some ecological literature where they're like a cottontail ate a bug. But one of the interesting things that cottontails and jackrabbits do, they do this thing called psychophagy, where they actually eat stuff, kind of partially digest it, poop it out, and then eat it again. So they might be, in a way, there's actually one individual that was super high in nitrogen, one of those lagomorphs, I think from hummingbird, or I forget where actually, but there's really high. And I was like, what is going on with that bunny? Like, why is it so? It looks like it's a straight up carnivore. Yeah. And so I was thinking, like, how would psychophagy or coprophagy is different if it's actually just straight up waste. It's totally like it digested, but they eat it anyway. I always think about how that could impact nitrogen values. Nitrogen values are super complex. I won't get too far into this, but it depends on diet quality type. It depends on body condition, that animals, how far they occur. If you're starving, you start eating yourself and you'll be higher in nitrogen space. So there's all this kind of stuff that you have to think about. So it gets complex.
Interesting. Okay.
So there should be a little bit of overlap.
So there could be some self-enriching.
Yeah.
We'll just call it self-enriching of your nitrogen isotope ratios. It'll leave it at that. Here's a question that is just about something that I've never heard of. So if you've never heard of it either, no problem.
Okay.
But I've never heard of it. Mark asked, he said he was sure other people would ask about this. Can you explain the concept of protein poisoning, which is also called rabbit starvation? Have you ever heard of something called rabbit starvation or protein poisoning?
Oh, no.
Oh, man. Mark, you're gonna have to write us an email about that one.
Yeah.
Jonathan Dombrosky, look him up, write him an email, give us some citations. It piqued my interest too. Copy me on it. Rabbit starvation or protein poisoning. Somebody who knows?
Yeah, I'm sorry, I don't know.
No, it's all good. So Winston asked if you or other if you or others have investigated the ethnographic record on this subject, is the evidence or information you presented here supported in the ethnographic record from traditional or native peoples, or maybe too much time has passed such that this information is not well maintained into the present? So
there's definitely an ethnographic record of it. That Stevenson Jordet papers, ethnographic. There's an ethnohistoric, of pest management and taking rabbits. But what I'm trying to kind of say here is this opportunistic thing of, oh, we just manage some pests and that's garden hunting. I don't know if I really buy that. Garden hunting is a specific subsistence mechanism that needs to be used, and we need to be certain about what it is and what it's not. So again, I'm not saying that that things weren't hunted in gardens, but if garden hunting is happening, it should be the majority of the signal that we see, right? It's the difference between managing a field for pests and viewing your field as a protein trap. And I don't think that's a little thing to just assume personally, but I get geeky about this stuff.
Yeah.
So there is ethnographic evidence of bunnies being hunted in fields for sure.
There were a couple of people that had questions just about the range in carbon isotope ratios. You talked about you showed that one sort of toy graph at the beginning, which was really useful. That you showed the tree as a C4 representing C3 plants.
Right.
You showed corn and grasses, dryland C4 plants.
Right.
And then you showed, later you showed humans show up over there looking like they're eating a lot of corn.
A lot.
A lot of corn. But we hear about people eating corn, beans and squash, and certainly people ate a lot of other things. Why? Can you tease that apart a little bit?
Yeah.
It might have sounded to some people like you were saying they were totally eating corn.
So that's a great question. We're mixtures of what we're eating, right? And so if you eat a little bit of something or a moderate amount of something, but you're eating a hell of a lot of something else, that signal will get swamped out. And that's basically the idea of why humans occur in that C4 space. It's the majority of the diet is C4. I mean, there's just no arguing that. But of course they ate other different things, right? Beans and squash. It's just not in the same amount. And it's the amount, the mixture. And so there's actually a brand new paper came out on this the other day by Paul Spock, who was also on that paper by Eric Gary.
Yeah.
There's a whole technique in stable isotope analysis called mixing models, where you actually take proportions of diets, you put in this crazy modeling, proportions of diets of different things, and then the values of those things. And then you can try to back calculate out percentages that there were in the diet. So you could make, but it takes a lot of assumptions to do that. But we could basically come up with a calculation of percentage of how much beans were in the diet, maybe, but it would be really low.
Got it.
But they're there. It's important.
People are interested. You focused on bones in yours, and so most of it was, I guess, isotopes from collagen.
Yes, it was collagen. To be clear about that was all collagen.
Can people? Nancy Nyberg asked about rabbit skin blankets.
Ooh.
Have there been any isotope studies that you know of rabbit skin blankets?
Wow.
And that sort of thing.
Man, that would be so interesting to look at. I don't know of any. Getting permission to do destructive analysis on a rabbit skin blanket would be a little tough. I'm sure maybe, on a small sample, you could get something. But what's really cool there, to kind of build off of her question a little bit, is you could be looking at, so collagen is incorporating over a longer time period of the diet signal, right? And keratin, hair, is a quicker turnaround time. So you could compare the difference between these quicker kind of tissues, hair, to the to the bone. So you could ask maybe a more specific kind of question and say, maybe like Goldstein was getting at was like, maybe that bunny got really risk adverse and was just like, I'm going to start going in that field now. And maybe you could pick that up in hair, get a more, a tighter.
Got a harebrained idea. Yeah.
I hope that makes sense. But still, my point with the collagen still rests.
Yeah.
If it's going to be the majority of the signal, there's got to be a baseline level of predation allowed on crops.
Got it. It also weighs on the question of domestication of rabbits, which I haven't really heard discussed. And you're right. Maybe we should be we should be wary of that discussion.
I've talked to people who have been like, rabbits were being domesticated in the Southwest, and I'm like, no.
Well, I want you to get permission to do isotope analysis on rabbit blankets, because if you think of turkey as right. A prototype for how domestication has happened in the Southwest, we see turkeys being domesticated for there's more economic uses, right? For blankets, turkey feather blankets, for ritual, and so it would be interesting to know whether you see a different C4 signature in these rabbit fur blankets.
That would be crazy. That'd be cool.
Wild. All right. Well, listen, that about wraps it up. I want to thank you for a wonderful presentation today and a lively discussion. I really enjoyed it, and I hope everybody else did too. Have a look, Jonathan. Have a look at the chat in the on Facebook as well, because people are always really laudatory.
Becca said, "Nice beard."
Yeah, nice. Becca said, "Nice beard." It is a nice beard. Everybody, thank you so much for joining us today. Be sure to join us in 2 weeks on December 3rd for Todd Suravel's discussion. And that one's co-hosted with the Pueblo Archaeological Society. Really exciting to be co-hosting with them again. And have a safe and enjoyable Thanksgiving. Thank you all, and be well.
Yep. Thanks, everyone.
Bye.